Okay, so looking at the topic of genetics and looking at doing monohybrid crosses, the first thing we need to know is that every mammal has two sets of chromosomes. So they're called homologous chromosomes because you get one set from your mother and one set from your father, so you always have two of the same chromosome. So this chromosome will have all of the genes for exactly the same traits, but it came from the mother, and this gene will have exactly the same, but it came from the father. So on each point, that gene for that specific trait will be the same trait. So for example, hair color or eye color or anything like that will be in the same place, but on two different chromosomes. And because of that, we can have what we call alleles. So alleles are all of the different forms of the same gene. So I have one allele here and one allele here. So that means for every single cross that I'm going to look at what my potential genotypes and phenotypes will be, I can have two different alleles that I'm going to represent, and they're either going to be dominant, okay, and a dominant allele is always expressed, and we represent it with a capital letter. So if there is a dominant one here, that is going to be the gene that you are going to have, that will be the trait. If it's recessive, that means it's only expressed when there's another recessive. So both of those need to be recessive genes for you to have that recessive trait, and that will be with a small letter. So for example, if I have dominant gene being red flowers, and this one says red, but this one doesn't, the flowers will still be red because there's one dominant gene. If the recessive gene is for white flowers, this one and this one both have to say white flowers in order for it to be recessive. So, because we have two different alleles for each of our genes, we can have different combinations. So if I have a capital letter for A, I'm going to say that is the dominant gene, and I can assign then the small letter for the recessive gene. So we always use the same letter to represent a gene. One will be capital, one will be small. So you always try and find letters that look different when capital and when small, so we know which is dominant and which is recessive. And now if I put them together, because remember there's going to be two at each time, I could either have the same for both. Okay, so if those are the same, that means that those are going to be what we call homozygous. So homozygous always means the same. Homo, same, zygous, genes. So we've got the same genes, and these are capital letters, so they are called dominant. So we've got homozygous, dominant. If I have both small letters, again they're the same, so that will be homozygous, but now they're small letters, so it's homozygous recessive. And then if I have different, so now I have a capital and a small, because they're different, now we call them heterozygous. So now we have heterozygous genes because we have one big and one small, so they are different. So hetero means different, zygous is doing with the genes. So all of these letters that I have, that represents what I call a genotype. So the genotype of anything is the specific genetic makeup in terms of our letters. So I will say that capital A is the genotype for an example of black hair. So I know that black hair is the dominant gene, and capital A is its genotype. The black hair part would be what we call a phenotype. So a phenotype will be exactly what you're looking for. So the phenotype, for example, if it's dominant, would be black hair. And then I can say maybe the recessive gene is brown hair. So the phenotype for the recessive would be brown hair. And then if it's heterozygous, remember, as soon as there's a dominant allele, that's the one that's always expressed. So big A, small a. If black hair is dominant over brown hair, this one will mean its phenotype still has black hair. Because we're always going to have dominant genes being expressed. So the phenotype will be what gets expressed physically, and the genotype is what the genetic makeup is. So if we look at some examples of what types of questions you get for genetic crosses, you have an example here where we've got, in Mendel's hybridization experiments, he crossed plants that were homozygous dominant for purple flowers, and they gave it a capital A, with plants that were recessive for white flowers, and he gave that a small a. The image below shows the ratios of the F1 and F2 generations of offspring. So in genetic crossing, we always start with what we call the parent generation, and when those get crossed, we end up with our first generation, the F1, and then I can take some of those from that generation and cross them again, and that becomes the F2 generation. Okay, so we know that now our purple flowers are the dominant ones and white flowers are the recessive ones. So the first thing they want you to do is state the genotype of the plant with white flowers in a parental generation. Okay, so for white flowers, it is recessive. So in order for it to be white, both of them have to be recessive, remember, because we can only express that when both are small letters. So the white one's genotype, remember the genotypes are going to be letters, so that would be small a, because it is homozygous recessive. So that would be your genotype. Then they want you to draw a Punnett diagram to illustrate the cross shown at the F1 generation and give the genotypic results of the F1. Okay, of the F2, sorry. So a Punnett diagram is always taking our letters and then we put them into what we call a Punnett square to show the crosses. So we're starting with F1. So F1 was taken from that cross and that cross from the parents. So the recessive one, we know, was small a, okay, because it was recessive. And it says the purple flower that he started with was homozygous dominant. So homozygous, they were both the same with capital letters. So if we have to show a Punnett square for that first cross, what we will do is we will take our parent generation and our parent genes were small a, and they were being crossed with big A. So your first step is to write what we call the gametes. So the gametes are when each of these genes is going to separate during meiosis. So remember, we're going to split during meiosis, and that would go into one cell, and that would go into another cell. And then the same would occur on the other side. So we end up with 4 different cells with 4 different genes through random assortment, and those would be my gametes. So now I can take those gametes and I can put them into what we call a Punnett square. So I take the first ones from my one parent, and that was small a and small a. And then I take my second ones from my second parent down the side for big A, big A. And now I cross each one in the square. So I start with A, so my first row would have an A, and then I go down by column for small a. Then I go across, that's big A, and I go down for small a. Again, big A down for small a, and big A down for small a. So your Punnett square will always have 4 options in it. In this case, all of them were big A, small a genotypes. Which means, if I look at the phenotype, they are all purple for the phenotype. Which is exactly what that says here. So they have all plants were purple. But now I know that this is the genotype for these purple plants. So this one was big A, small a for all of them. So now my question asks us to do a Punnett square for the F1 generation being crossed to make F2. So I'm taking one of these and one of these to cross. So my F1 parents were big A, small a crossed with big A, small a. Okay, so now my first step is to get the gametes, and I say each one in its own cell when I split them. So now I've got all of my different single combinations and all the ways that they can combine in a Punnett square. I put one parent at the top and one parent down the side, and then I cross them. So going across the rows, I have big A, and I go down the column, big A. Across the row, big A, down the column, small a. Across and down, big A, small a. It just always looks nicer to write the capital first, and then down and across, small a, small a. So those are all my possible offspring that I can have. So my F2 genotypes are going to be 1, big A, for that one. Then I've got 2 big A, small a's. And then I've got 1 small a. So those are all of my possible genotypes, which means that I've got, if I want to do it in a ratio of 100%, 25%, because remember it's out of 4, to 50%, to 25%. We divided that out into 4s. Okay, and then the phenotypes of that, what they're going to look like, is big A would be purple because it's dominant. Big A, small a would also be purple because it has a dominant one. And small a would be white. So there I have 1 purple plus 2 purple. So I would have 3 purple to just 1 white one. And another way to say that is 75% to 25% for our ratio. So there, they've only asked for the genotypic results, but it is always nice to write both. So you've got your genotypes at one at the end and the phenotypes. So that would be how we draw a Punnett square. So if we look at another example, I have got the mutant allele for sickle cell anemia is recessive. You need to draw a genetic cross or a Punnett square to determine the probability of parents who are both heterozygous for sickle cell anemia having a child with sickle cell anemia and then you need to show the ratios of the phenotypes arising from the cross. And now they give you a key. So they say capital letter must represent normal and small letter must represent sickle cell. So the first thing we have to do is we need to work out our parent genotypes. So we've got the parents who are heterozygous. Okay, so heterozygous means they're going to have one capital and one small. So that means it's going to be capital N, small n, being crossed with capital N, small n, because they're both heterozygous. So now I can look at gametes, and I can say each one gets its own cell. And then I'm going to draw my Punnett square. So in my Punnett square, I take my first parent, and then I take my second parent down the other side, and now I need to cross them. So I go across my row, and I've got capital N, and then down my column, capital N. Across my row, capital N, down my column, again with my column and my row, and my last one. So those are all my possible combinations that I have for my offspring. So now my offspring genotypes are going to be exactly what I have in here. So I have capital N, capital N. So there's one of those. Then I have capital N, small n, and there's two of those. And one small n, small n. So those are all four genotypes together. And what that means for phenotypes, so what they're going to look like, is that those all have dominant genes. So dominant genes means that they are normal, okay? Because it tells us that sickle cell anemia is recessive. So the capital N's means normal. So there are 3 that are normal with no sickle cell. And then just 1 that is recessive. So that means one of them will be recessive and one will have sickle cell. And if you want to write that as percentage as well, that would mean out of those 4, 75% have no sickle cell and 25% would have sickle cell. So that is then our phenotypic ratio, which is what we were asked for. So in any question, all you have to do is, if a letter is not assigned already, you assign a letter, capital for dominant, small letter for recessive, and then you always check, are our parents heterozygous or homozygous? Heterozygous, they're going to be different. Homozygous, they're going to be the same. And then we follow the steps for a genetic cross. Then the other options that we could get for questions is sometimes we specifically talk about genes that are sex-linked. So we have genes, just one pair, the very last pair, are your sex chromosomes. So your sex chromosomes are the only pair that are different for males and females. So the sex chromosomes in females would be capital X for the females. Males have a capital X, Y. Okay, because if you looked at the chromosomes in real life, you will see that one of the chromosomes is slightly smaller, so we call it the Y chromosome. Now, when we have genes that are sex-linked, so there can be specific traits that are linked to these X chromosomes, they're not found on the Y because the Y is missing that extra branch. So all of our sex-linked chromosomes would be on the X, and the way we represent them is we say X, and whatever gene we're looking at, we write as a little superscript. So still capitals for dominant, small letters for recessive, but it's always going to be based on the X chromosome. So that could be X capital A and X small a for a female, but a male will only get one of them and then a Y. The males will always have the Y with nothing on it. Okay, so in this case, a male, if that is dominant, he will always have that trait. If that is recessive, he will always have that trait with just one chromosome, because there's only one option there. Whereas a female could have both, and if she has a recessive gene, she can be called a carrier of that gene, because she will look like the dominant trait, but she will be having a carrier gene. Because if a male then gets that trait, he will be the recessive trait. So if we look at an example, in fruit flies, the eye color genes are sex-linked. So now we know we're dealing with X's and Y's. Red eye color is dominant to white eye color. So use a genetic cross or a Punnett square to show the cross between a white-eyed female and a red-eyed male. Express the probability of the expected genotypes and of the offspring as a ratio, and use the following key in your answer. So red eyes must be X capital R and white eyes is X small r. So in my parent generation, I am starting off with a white-eyed female. So white eyes is X with a small r. So female has to be XX. So that's X small r, X small r, because she can only have two small r's to have the recessive gene. And then a male, so we're going to cross that with a male who is going to have red eyes. Now it doesn't matter to be homozygous or heterozygous for the male, because he can only have one X gene. And red eyes is capital R. So he has X, capital R, and then Y for a male. And that, remember, doesn't get assigned anything because it doesn't carry the gene. So now, in my gametes, I've got X small r, X small r, X capital R, and Y. So now I'm going to do my Punnett square. I'm going to take the one parent at the top and the one parent down the side. And now I'm going to cross them. So now I go across and I say X capital R, X small r. And I go across X capital R, X small r. And then if I'm going down, I've got X small r Y and X small r Y. So now those are all my options with X's and Y's. But the double X's are females. So that's a female and that's a female. And the XY is male. So now when I'm writing it down in terms of my genotypes and phenotypes, I need to write down whether they've got red or white eyes and whether they're male or female. So my genotypes are going to be X capital R, X small r, and those are both the same. So there's two of those. And X small r, Y, and there are two of those. Okay, so what that means for phenotype is X capital R, X small r, it has the dominant gene as well as the recessive gene. So the dominant gene is going to take preference, and we know that dominant is for red eyes. So this is going to be a red-eyed female. Okay, and X small r is going to be the recessive gene. So that's going to be white, because remember the Y doesn't have any effect. so that is going to be a white-eyed male. And now that was 2 and 2. So 2 and 2, if we simplify the ratio, would mean 1 to 1, because that's the simplest form that we want. So I'm going to have 1 red-eyed female and 1 white-eyed female. Or if you want to do that as percentage, 50-50. Okay, but it did ask for a ratio. So a ratio means numbers. So you keep it like that. Okay, so as soon as something's sex-linked, we've got to use X's and Y's. So if we look at one last example for sex-linked genes, here I've got hemophilia is caused by a recessive X-linked mutation which prevents the synthesis of factor 8 by the body. This condition will lead to uncontrolled bleeding and possibly death. Unless hemophiliacs receive injections of that factor 8, their blood will not be able to clot. So now, a carrier mother, which means she's heterozygous for hemophilia, and a normal healthy father decide to have a child. The mother falls pregnant with a son. What are the chances he will suffer from hemophilia? So it says you don't need to, but we're going to do this in terms of a Punnett square just to show you exactly what our steps are. So we start with our parents again. So now it's sex-linked, so we know we're going to have X's and Y's. Okay, so the mother is heterozygous for hemophilia. So we're dealing with hemophilia being recessive. So what we can do is we can use any letter that we want, but we have to say what they are. So let's say that our key X, capital H for dominant, and that would be normal. And then X, small h, for hemophilia, because that's recessive. So, the carrier mother is heterozygous, so she will have a capital H and a small h. But the father is normal and healthy, so just on his X, he has to be capital H because he's normal. And remember, the Y gets nothing. So now if we do the gametes, you're going to have X, capital H, X, small h, X, capital H, and Y for our different gametes. So now we can cross that in our Punnett square with our one parent on the top and our second parent down the side. And then all we do is we cross inside. So going across, I have X, capital H, and down the column, X, capital H. Going across to the next one, X, capital H, X, small h. And then going down, X, capital H, Y, and X, small h, Y. So now our offspring genotypes are going to be X, capital H, X, capital H, and there's one of those. Then I've got X, capital H, X, small h, and there's one of those. Then I've got X, capital H, Y, and there's one of those, and X, small h, Y. So there's one of those. So now the phenotypes. Remember, you always have to talk about male and female as well. So capital H means that those are both dominant. Okay, so that would be one normal female. Then capital H, small h would mean that they are a carrier. They are heterozygous. So they have the possibility to pass that gene. So we can say that that is one carrier female, but she is normal, not having hemophilia. Okay? And then capital X, capital H means normal with no hemophilia. So now that's the only gene that counts. So that will be one normal male and small h on the X means that that would be one hemophilia male. Okay, so now those are our females and those are our males. But our question asked, what are the chances that the son will have hemophilia? So out of the son, there's only two different options that we have for the males. So out of those males, one would be normal, one would have hemophilia. So that works out to a 50-50, just in terms of the males, because we already know that she's just having a son. So there is a 50% chance that he would have hemophilia for our final answer. If that was asking just in terms of children, what would be the percentage? That would obviously go down to 25% because there are 4 different options. But because it's just within the males, there's only one of each. So that's why we get to 50%. So those are the different types of examples you can get when you're required to do genetic crossings and use your Punnett squares to find out either you're going to use just normal letters for any sort of normal gene, or it can be sex-linked where you use your X's and Y's. But just remember your terms, homozygous being the same and heterozygous being different, and whether it's dominant or recessive. Because your dominant gets your capital letters and your recessive gets your small letters.